protein chemistry, till now we have done a few lectures and certain conceptual clearance, which I thought uh, may be useful. They are important, for the ones who are examination oriented from Viva, etc. But more importantly, they are important for your understanding. And if you understand a thing, then everything becomes very smooth. Otherwise, mugging up repeatedly, that leads to confusion. So in those lectures, when we talked about classification based on polarity and metabolic fate, etc., etc., so we seem to be obsessed about a basic set of 20 amino acids. And these were called standard amino acids and primary amino acids. Although more than 300 amino acids are present in our body, some of them occur in protein. For example, hydroxylysine, hydroxyproline, desmosine, hydroxylysine, hydroxyproline in collagen, desmosine in elastin. Then in certain, like for example, ornithine and citrulline, etc. You will learn about them later on. So these amino acids are present in free form. So more than 300 are present in the body. So what is so special about these 20 that we seem to be obsessed? The students often say that, because they are more predominant. Correct answer, they are more predominant, but there is no cutoff limit. For example, collagen ke andar, hydroxylysine, hydroxyproline are also present in large amounts. तो कोई कट ऑफ नहीं है कि इससे ज्यादा हो तो प्राइमरी. There is a better reason also, that is that they are specified by the genetic code. So they are DNA coded amino acids. जब आपने स्कूल में भी किया होगा तो जेनेटिक्स की थी. So you must have seen 4 raised to the power 3. There are 64 codons. Of these, 3 are stop codons. Left are 61. So 61 codons code for 20 amino acid. So this, if you remember, was called degeneracy of the genetic code. So 20 amino acid for which these 61 codons are encoding, they are called primary amino acid. Then there is certain arbitrariness also. For example, 2 amino acids linked together are forming dipeptide. ये है जैसे 1 amino acid, carboxyl group of 1, amino group of 1, elimination of water, and a CONH linkage called peptide linkage is formed. So 2 amino acids linked together called dipeptide. 3 amino acids linked together, tripeptide. Although there are only 2 peptide bonds, but we count number of amino acids. Now examples, glutathione, antioxidant part play करता है. TRH, thyrotropin releasing hormone, which helps thyroid hormone का ultimately release वगैरह. Then there are nonapeptides, oxytocin, ADH, etc., etc. So peptides, few peptides linked together, we call them oligopeptide. Many amino acid linked together, that is called polypeptide. So few is oligopeptide, many or several, that is polypeptide. Now there is arbitrariness, what we were talking about. It is mentioned that about 50 amino acid linked together, usko hum up to 50, we can few to 50, it is oligopeptide. और उसके बाद उससे बढ़ जाए तो it is polypeptide, but it is arbitrary. And when do you start calling a polypeptide a protein? When the molecular weight exceeds 10,000. 10,000. सो यहाँ पर एक इम्पॉर्टेंट बात जो हम करना इम्पॉर्टेंट होगा कि what is average molecular weight of an amino acid. If you count 20 amino acid का molecular weight divided by 20, it comes to 138. But considering a fact that the smaller amino acids are more predominant compared to the larger one. So this is called geometric mean, isn't it? So if you consider this point, the average molecular weight becomes 108. So that means 100 or more amino acid linked together, then you start calling it a protein. But this is again, I'm saying, arbitrariness. This is all arbitrary. Another feature of arbitrariness related to a non-essential amino acid called tyrosine. There was a classification scheme based on identity and structure of the R group. So this can find place in two categories. Number one, aromatic amino acid containing an aromatic ring. And it also contains a hydroxyl group. So this can also be subclassified as hydroxyl group containing amino acid. An important point, all these amino acid, for example, this amino acid we will call L amino acid. There is another category also D. For example, if it was like this, NH2, and here if it were H, then you would have called it D amino acid. Now an important statement that all amino acids in our body are of L variety. All amino acids are of L variety. All carbohydrates are of T variety. L and D, these are enantiomers of each other. And normally, you know, the students make this mistake. जब ये left पर होगा, then it is L. जब right पर होगा, then it is T. But please remember, it's not, we put it on the paper like this, but otherwise it's a three-dimensional structure. So we take glyceraldehyde as the reference compound. So this is L-glyceraldehyde, this is D-glyceraldehyde. They are mirror images of each other. Mirror images, as you can see. So L amino acid अगर आप देखिए तो ये L-glyceraldehyde से resemblance है, D की इससे है. And here it gives me opportunity to tell a very important point which you will study in enzymes. That is called enzyme specificity. Specificity. Enzymes are highly specific. For example, glucokinase, which converts glucose to glucose 6-phosphate, the first step in the glycolytic pathway, which commits glucose to the intracellular metabolism, etc., etc. This enzyme or the other one, hexokinase, will act only on D form of glucose. D और L में फर्क क्या है? Exactly same, except orientation different है, but the enzyme detects that also. Likewise, the enzymes of amino acid metabolism will act only on the L form of the amino acid. So यहाँ पर asymmetric कार्बन एटम का एक कॉन्सेप्ट है. एमाइनो एसिड के अंदर अगर आप देखिए ये this C, it has got 4 different groups attached to it. So wherever such carbon atoms are called asymmetric carbon atoms. Wherever asymmetric carbon atom is present, that imparts two important properties. One is this D and L, what we call stereoisomerism, and second is small d and small l. This is called optical activity. Optical activity. They have got no relation to each other whatsoever. So D or L हमने देखा कि they are mirror images. This small d and small l, now they are abbreviated as plus and minus. This is based on in which direction the plane polarized light is rotated by the solution. So optical rotation, L form, I have said, both these properties are imparted by asymmetric carbon atom plus right direction minus left direction, etc., etc. Now, an important thing, one amino acid does not show optical activity or stereoisomerism. That is the smallest amino acid that gives rise to largest number of products. Why it does not? Because it lacks asymmetric carbon atom. Why? Because asymmetric carbon atom is H. So you can see here two H's are attached. So this is not asymmetric. So it does not have D or L, or it does not show optical rotation. One more conceptual point. I think this I have. You can see some more examples also that we have discussed about tyrosine. Tyrosine can be called non-essential amino acid. Tyrosine can be called glucogenic and ketogenic, both sulfur containing. You can see some other examples also. Now, glucogenic, ketogenic की अगर हम बात करें, तो we call some amino acid as glucogenic because they can form glucose. So they can form pyruvate, they can form glycolytic intermediate, or they can form TCA intermediate. And you people know that these compounds are substrates for gluconeogenesis. They can form glucose. We took a very simple example last time, alanine. Alanine can form pyruvate, and pyruvate is a substrate for gluconeogenesis. We took example of aspartate. Aspartate can form oxaloacetate. A TCA intermediate can form glucose. We took example of glutamate, which can form alpha ketoglutarate, a TCA intermediate, again a substrate for uh, glucose. Ketogenic are those amino acids which yield acetyl coenzyme A. Now, a question may be asked that acetyl coenzyme A can also be converted to glucose, but the answer is no. And it is capital NO, no. Acetyl coenzyme A is not a substrate for gluconeogenesis because the reaction pyruvate to acetyl coenzyme A is irreversible. Opposite reaction cannot take place. So therefore, these amino acids are not capital NOT, not glucogenic. Rather, acetyl coenzyme A can form something called ketone bodies, which you will learn later on. So they are called ketogenic. And then glucogenic come ketogenic. Now, variability. Variability, proteins are most diverse molecules in our body. Most diverse. इतनी diversity किसी में नहीं होती है. Why? Because the variability resides in R groups and how R groups are arranged. Now, I'll give you a very simple example. You read it आगे से या पीछे से, it remains the same. But you read, can you make sense of this word? No. But Ulta Pado Modi, who does not know? For that matter, you might be knowing Dinesh Puri, but do you know Ripu Shanedi? It is Ulta if you read, it becomes. So even if you change a single word in a polypeptide chain, this may change the very identity of the protein. So the variability resides in sequence of amino acid, which we call primary structure. You are going to study later on. Variability hai. Just say, for example, if you take only 20 amino acid, and how many dipeptides you can construct out of them? 20 raised to the power 2. कितना हो जाएगा? 400. How many tripeptides you can construct? 400 multiplied by 20 कितना हो जाएगा? 16,000. I may be wrong. ठीक है? टेट्रापेप्टाइड कितने हो जाएंगे और कितने प्रोटीन्स हो जाएंगे? 20 raised to the power 100. A very mind boggling figure. So अगर सिर्फ प्राइमरी को ही कंसीडर करें, वेरिएबिलिटी इतनी enormous variability is there. Now, lastly, probably, time is moving on. Non-polypeptide portion of the proteins, they are called prosthetic group. For example, hemoglobin. We discussed last time, oxygen carrying molecule having enormous buffering capacity because it is very rich in the histidine, the amino acid, the only amino acid which acts as the buffer. Isn't it? और hemoglobin के अंदर the prosthetic group is called heme. And you will study later, heme is a 4 rings होते हैं और उन rings के बीच में होता है आपका iron. So iron at center of the ring is there, and it is the iron which binds with the oxygen. So hemoglobin, which is oxygen carrying molecule, तो ऑक्सीजन कैरी के लिए तो सिर्फ आयरन ही काफी था. सो व्हाई सच ए लार्ज मॉलिक्यूल? देयर आर 4 पॉलीपेप्टाइड चेन, अल्फा 1, अल्फा 2, बीटा 1, बीटा 2, देन अ टेट्रा, पायरोल, 4 रिंग्स और 4 रिंग्स के बाद बीच में आयरन. अगर आयरन ने ही कैरी करनी थी ऑक्सीजन, तो व्हाट वाज द नीड ऑफ ऑल दिस? इसका सिंपल रीजन है. इसका मैं आपको एग्जांपल से बताता हूं. Heme is present in cytochromes also. You must have seen in electron transport chain. Cytochromes के अंदर ये ऑक्सीजन कैरी नहीं करते. They are transmitting. They are involved in transmitting electrons. और हीमोग्लोबिन के अलावा हीमोग्लोबिन का cousin है myoglobin present in the muscle. This also binds with the oxygen, but the oxygen binding properties are entirely different, and you will study P450 cytochrome. This also has got this, heme as the prosthetic group. But क्या role ये करता है? This is involved in detoxification of drugs. So you can see the same heme is present in all these proteins, but it performs different roles. And what determines this different role? Non-polypeptide portion. This is also called, you will learn, apoprotein. So prosthetic group is important for actual performance of the function, but what function it will perform will depend on the non-polypeptide portion, that is also called apoprotein. So some conceptual clearance I have tried to give, some very commonly asked question, but more importantly, something important for your understanding. Thank you so much.